I'm amazed. When I woke up this morning and saw the weather forecast, I thought, whoa, there's going to be Jane, and I was persuaded Cornelia, my wife, to come along as well. I'm staggered that with the weather that we've got, that you're prepared to give up an hour of your time or longer to listen to me prattling on. But anyway, let's hope it's not wasted. So algae, art, aesthetics and architecture. I'm presuming nobody in the room will know anything about algae except Peter Williams, who's sitting here at the front, who knows a little bit, it must be said. Yeah. So I'm a scientist. I work with algae, and there's a big drive in the science that we do and the kind of stuff, my boss is my Pro-Vice Chancellor for Research. Big science and big science programs are what drives modern day science. And you'll hear about wonderful government initiatives, the new institutions and foundations that are being developed, and the big programs. And it's great stuff, and I really appreciate that, of course I do. But I like this slide a lot because actually behind every good scientist is somebody with some creativity, innovation, and ability to think outside of the box. So although this is the juggernaut that drives our lives at the moment of science, I find it very difficult. I don't think you can do that if you don't have the VW in front of it as well. And I think, hopefully by the end of it, this whole divide between art and science, it's long history, and everybody says you're either a scientist. Our school system kind of develops it, doesn't it? You either go into the arts and humanities or you become a scientist, and it's very difficult even when you're making your own GCSE choices. You've got to make this divide as to what you really are, and there's very few people that are able to kind of combine the two, and it's such a shame. This is really a really awful cartoon that was drawn for me by Peter Schreck, actually. Look at that, the artist is a woman and the scientist is a bloke. So it really is an awful stereotype picture. But I think there's initiatives, and they crop up every now and then, about, well, let's have art and science coming together. They're great, and I appreciate that. But I think we need it to be a much more bottom-up approach where both artists and scientists actually decide to cross that divide rather than it being some kind of funding scheme that makes it kind of possible. So the starting point, there's this guy, Poincaré, and you'll see this quote all over the place, and it's quite often misquoted, as most quotes are. So the scientist does not study nature because it's useful, he studies it because he delights in it, and he delights in it because it's beautiful. And that's normally where it stops. And everybody says, well, isn't that great? And it's in the beginning of books, and I think I've even used it in the beginning of a book as well. But he then goes on to say, and what people leave off is, of course, I do not here speak of that beauty that strikes the senses, so the aesthetics, he's not talking about that. He's meaning that profounder beauty which comes from the harmonious order of the part. Remember, he was a mathematician, and he dealt in mathematics, and he was looking at the order of numbers, and he was looking at the way things were formed. And that is, I guess, the link that I'm trying to bring out at various stages of today's talk. Great quote at the top. That needed to be kind of, the second part of the quote needs to be used. Just to show that it's quite mundane, or can be quite mundane. We get new tranches, we get 125 new marine science students every year. I kind of talk to them at the beginning of when I give the head of school welcome, is that their greatest tool that they have, we've got lots of very sophisticated equipment. We've got ships, we've got bits of kit that cost 50K, 100K, massive amounts of money. The ship I go to in the Antarctic, costs us £350,000 a day just to keep it on the water. So we've got lots of money being invested in science, but actually the greatest observational tool that any science has is their power of observation, normally their eyes, their senses, their smell, and touch as well. So you can even see, even when cutting vegetables, so celery and red cabbages are my favorite forms where you see the patterning. The whole thing is designed carefully to package the celery or the red cabbage in a perfect way, so it's the least damaged and actually functions as a vegetable properly. So it's not there just at random. So when you kind of deconstruct that a little bit and put some more meaning, is you can see the spirals. And you can see the patterning that is there, and you can even eat it afterwards as well. So power of observation, even in the most basic of things, is absolutely key. You even see it in the famous ink blot designs. So, you know, dropping ink from different heights. So this is quite close and further away. And you see the way that you get these organic shapes beginning to form. So this is just dropping ink onto a piece of paper from different heights. And these resemble microorganisms that you see under the microscope. So they resemble bacteria when you kind of get into this stage. You know, pseudopodia and protrusions kind of creeping out. And so even that, tell your wife first, because I remember doing this on Christmas at the kitchen table, this experiment, because I read about it and I thought it was quite nice. And it's quite messy when you're dropping ink from a height. So do convey to your wife that you're doing it. And then even in broken windows. So this whole concept of this was a neighbour's window that cracked in one winter. But there's even, when you deconstruct this mess of the cracking and actually look at the shapes and actually the fractal, the mathematical patterns. So the stone hit here. If you ever have the misfortune of actually looking at a windscreen of a car when it cracks, before you actually get it replaced, maybe you should look at the patterns that are created within it. I mean, they are fascinating, and there is a mathematical form and fractal development that comes from something that would apparently be quite chaotic, or would seem at first sight to be quite chaotic. So as was said, I spent quite a lot of time in the Antarctic looking. I study how organisms live in ice. So when the seawater freezes over, organisms get caught into them, small microscopic organisms, bacteria and phytoplankton, and I study how they live there. But actually, as the ship breaks through the ice, the best place to be on the ship at any stage is on the bridge, looking down, and then you can actually see. And the ice breaks apart in fractal patterns that are very similar to what you saw, what I showed you there with the broken glass plane of our landlord's conservatory. And so throughout nature, whether it's ice, whether it's cabbages, whether it's celery, remember ice, it's millions of square kilometers of this stuff. At any one time, there's about 30 million square kilometers, area much bigger than the United States of America, covered in this that is breaking apart and jostling in these fractal patterns. So if you just kind of look, you see patterns everywhere, and that can be inspirational. And that's basically, I guess, this kind of stuff is what Poincaré was trying to say in words. As a mathematician, he would do the maths about the way this is breaking up. So, as a quick introduction, I'm going to end this introduction. There was this guy, Endel, in about 1898, who wrote in this Decorative Kunst, quite an influential guy in his own way. In the design movement and of the time. And he kind of came out with this: you must learn to look at a tree or a root or a stem or a leaf and see its form. So this whole concept of form and function is when this was beginning to become kind of talked about in and of itself. So, you know, and then to feel any changes in that form. So this kind of perception. So this is kind of one step on from where I guess Poincaré was actually trying. And if we learn to look at nature in this way, a totally new and unknown universe emerges. And I think he was just trying to stimulate in this phrase as to kind of actually step back and actually take a look. And this is the power of forms. They awaken our inner feelings. We all know what, looking at a coral reef, there's certain amounts of inner feelings. Looking at waves and the way waves break on the shore or the way waves break out at sea. There's inner feelings and directly, and it's not necessary to posit any other psychological process between forms and feelings. So this kind of, where I've taken you in this really very brief introduction is, the mathematics behind all it is, whether it's fractals of an ice floe breaking up or a window breaking, through to the way the process has on our feelings. So that's where we got. So that's how I'm kind of, if you like, where I hope to get to by the end of the main part of the talk, is to try and tie all of what I just said to you back together again. So back to algae and art. What are algae? So algae, so you'll know them all from the seaweeds that you see on a shore. They're called macroalgae, large algae. That's what I started studying as a PhD student or as an undergraduate. Actually, it was seaweeds that got me into science. I mean, you just thought, you know, the humble seaweed. So normally slimy, ugly, messy, inconvenient kind of stuff. I hope you'll come away from this seeing that there's something different. But also the phytoplankton. So these are the microalgae. These are the things that sometime in late April, early May, that if you look at the sea, especially if you look at the sea outside Menai Bridge, or the sea that you can actually see. So one night it will be crystal clear water, and the next morning you will wake up and you'll see that it's a muddy brown kind of coloration. This is called the Phytoplankton Bloom. It happens every year. These are the food. It's an explosion of growth of these unicellular organisms that divide very, very quickly when the temperature and the light conditions are right. And they're the basis, if you like, of the food chain in marine systems. So that's ultimately their food for small crustaceans, small organisms that are then eaten by the fish and so on and so on. So they're really very important. As such, not very many things eat seaweeds, but seaweeds have a lot of things growing on top of them that lots of things eat as well, and seaweeds are very important for coastal habitat. So I'm going to talk about stuff you can't see, at least in this form, without the aid of a microscope, and things that you will have seen if you visited any shoreline as well. So I like showing this because I'm a pedant, and it would be good if somebody in the audience remembers this. They're not plants. They are absolutely not plants. They look like plants and they act like plants, but they're not plants. So here we have an infrared picture taken of just outside of the lab in Menai Bridge, just on the Menai Strait. And what it is the fluorescence or the heat energy that is being released due to photosynthesis taking place is actually picked up by the camera. So basically the excess heat, if you want, is being, so it's basically where it's red, something's photosynthesizing, which is a very plant-like trait. So all these seaweeds are doing it, and you can see the trees in the background on the other side of the Menai Bridge are doing it as well. So you say, well, of course they're plants, David. They contain chlorophyll and they photosynthesize. That's as far as they get to for being plants. They use nutrients. They need nitrogen, phosphorus, iron, vitamins, and all sorts of other things, just like plants do as well. Plants get them through the roots, generally. Seaweeds don't have roots, but seaweeds are bathed, or algae are bathed continually in seawater, or at least when the tide comes in, the seaweeds are covered by seawater, and they're able to take it across most of their structures. So they don't have root systems, and they don't have internal transport systems for moving water and nutrients around to feed the leaves and everything else. No seaweeds have flowers, and I've covered the roots as well. So yeah, they're plant-like, they act like plants, but they're not. Onto the seaweeds, I think, again, if you stand back and look at the form, so there's a typical low, there's an aesthetic of that brown algae. It is slimy. It will smell if it starts decomposing, but in that form. This is something that has been left in that form. It's a huge brown filamentous algae and a green algae that has been left as the tide has gone on. You can see what the form is. But I guess with seaweeds, it's actually when you get in the water and you actually snorkel or dive or just stick your head under the water and have a look up, that you actually really get a sense of the beauty of what seaweeds are. Because when they're moving around with the tidal movement or with the current, they really are. There is a wonderful ethereal beauty that comes about it. It's quite amazing. It's better than a forest that is moving in the wind, because the water motion and the viscosity of the water and the way things move in water adds a whole different, if you like, dimension to that movement, and the way the form of the seaweed is translated to move ideally within that water body. Plants on land, forests, are quite clunky. There's things in the sea are really not clunky. I don't think you could say that at all. There is an appeal in the colour. So, I mean, a lot of the appeal comes just in the vivid and the variety, diversity of colours. And I've given this talk a couple of times in other places, and I've kind of had pictures of all the colours, but actually it looks a bit messy and it looks a bit over the top. But when you get these vibrant, bright emerald greens that just hit you straight between the eyes, and these really sophisticated reds and browns as well. So, of course, just like in anything, I mean, colour is vital to the enhancement of what you are seeing. But I guess with the seaweeds, and these are all UK seaweeds that I've shown you up to now, sometimes it's just a weird and wonderful form. So you've got this species of brown seaweed. You don't see it very often. It grows very low down on the shore, only very occasionally exposed at low tide. But you see these wonderful organic convolutions, if you want, that is actually vital. You know, this is generation after generation. This isn't some kind of abnormation, or this is generation after generation. This species of seaweed has evolved to have these convolutions, and I'm going to go into why that could possibly be. And then you have these other. This other kind of species. This is from Triada Bay up in Anglesey, which is just like a blob, but it's like a brain-like structure. And it's if you walk past it on a beach, you just go yuck. But actually, when you get close into it, it is amazingly the way over evolutionary time this beast has formed these pockets and these envelopes and it is quite a remarkable thing. So why would species of seaweed have these, if you like, weird and wonderful adaptations? This is a piece of seaweed, Macrocystis, taken off the Californian coast. And you can see, it's a massive beast. So it's stuck to the seafloor, and it can be stuck to the seafloor 30 or 40 meters away from where these blades, these leaf-like structures, float on the ocean surface. So it's a massive, long set of leaf-like processes which can be 30, 40 meters below where it's attached to the seafloor. And you can see these patinations in the blades, and you think, well, why on earth would evolution come about with this beast to kind of have this patinated leaf formation, which is really quite pleasing to see. The reason and it's physics. It's physics and math. So if you have a flat surface in a body of water, and you have water flowing over that flat surface, there's if you like, a dead zone. We call it a boundary layer, but there's a zone above that flat surface where things can't move backwards and forwards. There's no diffusion possible. So nutrients and waste products are just kind of stagnant there. So it happens in sediments, or if you put an oil rig in the middle of the sea, or if you put your hand there and leave it there long enough, there will be this dead zone. It can be a few millimeters, can be centimeters, depending on how big the thing is. But by having all of these undulations and these wave-like structures within the blade of the seaweed, that dead zone is disrupted. So it's not a flat layer, but it's a kind of an undulating layer with waves. And so that means that the boundary layer never actually forms, and so that the exchange of gases and nutrients, the nitrogen, the phosphorus, and the trace elements I've mentioned before, are actually able to take place. So there's a real function between this aesthetically pleasing, undulating structure within these seaweeds. And that's true for just about anything. These blobs at the end, that's what's filled with the gas that holds these leaf-like structures up on the surface of the water. So the physics of boundary layers, and there's a whole host of people that study this in great detail, and there's masses of equations and all the rest of it, is what has led to this aesthetically pleasing structure in the ice. Now, by the way, I am not an artist in any way. I study bacteria and ice. But there are a key number of people throughout history, tends to be biologists, who have that have artistic tendencies, let's put it that way, that have been able to jump this. And one of the key ones that you'll come across is a guy called, if you take it any further, there's a guy called T.A. Stevenson, who really had a passion for looking at things that people find on the seafloor or in the intertidal zone, and actually trying to depict them in a way that was aesthetically pleasing but realistic. That's actually quite a key thing to keep in your mind. He was actually realistically, so he took sea anemones, again globular glutinous blobs, but actually looked at them under low power microscopes and was able to show what they looked like. I mean, one of his most famous pictures is this one from 1934. This is a picture of anemones it is just mind-boggling. This is a glob of goo that he found on the shore, took his time to actually look under a decent microscope, and this is actually what was revealed to him. And Stevenson's books, they're becoming quite difficult to get hold of. You can still get hold of them from secondhand booksellers, and they are just remarkable. But his link with Poincaré was quite striking. He looked at the way seaweeds flop down when the tide goes out, and the way when the water leaves them, they just kind of, and you would imagine that would be quite random, but actually it's not true. A form of a seaweed is quite closely designed by which it's going to flop in such a way that certain fronds or leaf-like structures of the seaweed actually are on the top and are still able to photosynthesize in the light. So he looked at that in quite detail and was able to convert pictures like this into quite sophisticated mathematical ideas and sophisticated mathematical concepts. And he was able to kind of look at that, and he was able to convert a piece of seaweed with some snails grazing at it and the kind of geometry that is lying behind the way that the seaweed is as it is. He got into, so he was into anemones, he was into seaweeds. He was also into starfish and brittle stars that you commonly find in rock pools and also on shallow benthic habitats, as well as deep habitats, but you can easily get hold of brittle stars. So his work was fine. It must be said that he saw pattern and design in lots of things, and his wife was clearly a feature. So in all of his marine biology books, his wife suddenly appears in the frontispiece, and so does his car as well. So he was able. He had a broad kind of vision of what life is, and that's great. You'll see seaweeds, in the William Morris wallpapers and fabrics. You'll see seaweeds appearing, and they were really quite important, but they obviously weren't brave enough to just leave it at seaweed, so they had to embellish it with cornflowers as well. But it was clear that seaweeds do appear in a lot of really quite commonly viewed wallpaper coverings, and you can see them here as well. So there's seaweeds here, but they were never brave enough to just leave it at the seaweeds. They always put some terrestrial plants in to mix in with it. And of course, we've got Matisse, who drew seaweeds a lot. I mean, in the 1950s, this was actually called the algae. So I mean, it looks much like a lot of other Matisse work as well, it must be said to the untrained eye. But this is one that he actually did take inspiration in this cutout form for algae as well. Okay, so that's seaweeds, and so on to the microscale kind of thing. So this is pictures that I showed you before, things that look like this. These are things called diatoms. I'm going to come on to what a diatom is. And there's such a huge range of different types of form, type, shape, capability, whether they photosynthesize or don't photosynthesize. Some of them actually eat other organisms for some of the year and then don't for other times of the year. They are a most incredible group of organisms, types to actually study. And actually, when I said what got me into seaweeds in the first place, so my career, so it was Jacques Cousteau that taught me about marine biology, that there was such a thing about marine biologists. Seaweeds was an early career thing that kept me in the game, but it was during a practical as a second-year undergraduate when I looked down the microscope and saw something like that, I guess. Well, actually, I probably saw something more like that, but I imagined that. And that's when I decided to make a career in marine biology. So whenever I see this image, I think, yeah, as a second-year undergraduate, looking down the microscope for the first time, that was it. That was when I kind of said, that's what I want to study. If you look at them under a special microscope, a scanning electron microscope, this is what you see. So you see that kind of thing that the light microscope can pick up, but you actually see these patterns and these spirals and these holes and these plates that go up to make what we see refracting and reflecting the light. This is what you see in more detail. And I guess that is where we start getting closer to the mathematical stuff that is underlying everything and bringing it into what we see as aesthetically being pleasing. Okay, so diatoms. So diatoms are a group of phytoplankton, and they're the ones that, you know, when I said that during the spring bloom, those are generally the ones that start the spring bloom. When the conditions of temperature are right, they kind of go like that, and they start growing like nobody's business and dividing. Diatoms are quite fascinating. They are basically a plant cell, if you like, or a similar plant-like cell, so a chlorophyll-containing cell that is surrounded in glass. It's not quite right, but it's for all intents and purposes is correct. So a glass shell. And so if you look at it, if you look at this, this top picture here, this is a computer simulation of a diatom. So it's basically a container, and then a lid of it. Both parts of those containers are glass. Well, but of course I said that things like nitrogen, phosphorus, gases are important for these organisms, so they've got to be able to go backwards and forwards. And so there's lots of holes in these glass shells. And that's where the transport of material that is vital for that life takes place. So they are remarkably unique. And that patternation of those holes can actually, it's what taxonomists and people who are interested in what makes a species or defines a species have used, is the patternation of holes within those glass shells. Before we had modern molecular techniques, is what largely determined the taxonomy of diatoms. This is just interesting, is that the shape and the size of the holes can change. So this is one, what the type species looks like. This is what this particular species, but you put it into different saltiness of water. So this is one that has been put into salt water that is about half the amount of normal seawater salt. And you can see that the holes change in the shape and are impacted environmentally as well. So this is where all the gas and the nutrient exchange takes place. And actually just the study of how a hole forms in a diatom, it just shows what type of person I is. The shape of the holes, I mean, they are just stunningly fascinating. This is one that I kind of looked at for this piece, and then you kind of look at these organic shapes that come out, and as I say, environment is having an input into them. There's the inherent physiology or the genetic makeup of the beast itself is having an input. And yeah, I'm sure Matisse, if he'd had the opportunity, would have come up with something similar to this as well. And it's not just in diatoms, there's other microorganisms that are there. This is a group of things that you can actually see. They're very small, but they're about 1 millimeter in diameter. Foraminifera, you see them quite often on sediments. But actually, their mouth parts look very much, or the openings into the cell look very much like these holes in diatoms as well. So why am I prattling on about diatoms quite so much? Their shells are incredibly powerful. I said they're made of glass. Intuitively, I guess like I would have thought, well, that's quite fragile. But actually, when you actually look at the structures of these diatoms, they are incredibly tough. So this is a group from Germany at the Alfred Wegener Institute who have mathematically modeled them. So they've made the measurements of diatoms, they've taken mathematical models, and they have computer-based models that they're able to test. And they also take diatom shells, dead ones, and they kind of try and crush them under the microscope. So they take a diatom shell, they get rid of all its contents, and they're able to look at the loading of what it takes to break one of these things. And it comes out at about, to break this one, to actually distend it, is about 36 nanonewtons, well, meaningless number to me and to you, I guess. But the way that my colleagues explain it is that, well, that's the equivalent kind of force that is needed for you or I to break open a Brazil nut with our teeth. Well, most of our teeth would crumble and give way. So this glass shell is really very, very tough, and that's why diatoms are partly so successful. But lots of things, I told you a few minutes ago, that lots of things eat diatoms. And what has been really interesting is in the literature that I'm not following quite as much anymore, but I was until the point that I was able to follow it. There's been an arms war between the organisms that eat diatoms and the diatoms themselves over tens of thousands of years. So as organisms have changed their mandibles and their eating parts of in their jaws and also in their gizzards, lots of marine organisms actually break open things when they start getting into the stomach and their food passages. And the way they do it is they have silica nodules and silica armored teeth and silica armored mandibles. And these have got stronger and stronger and stronger, and at the same time as the grazers have developed their mouth parts and their stomachs to be able to crush diatoms, the diatoms have got thicker and thicker shells. So it's kind of called this evolutionary arms war in the sea. I thought, well, that's, especially knowing my colleagues who came up with this theory, I just thought, well, yeah, it's a guy called Victor Smetacek who Peter will know. Great, but actually the same thing's been going on terrestrial systems. So if you rub your hand down a piece of grass, it feels really very rough. And the reason it feels rough is because there's silica in the outside cells of most grass species. And there's been an equivalent arms war going on between grazers of grass on land and things like rabbits. Because as the grasses have put more and more silica in there, of course they wear away the teeth of the things that are grazing on the grass. And so there's an evolutionary race between the amount of silica in grass and the amount of the way teeth are able to be stronger or regenerate in the case of some mammals as well. So it's just intriguing. So these diatoms are really tough. They're difficult to eat, but they are eaten. This has led to some design going on. So these colleagues have had a lot of work together with some decent people to looking how diatom structure can be used for alloy wheel design. They haven't quite got to this point, but who knows? One day, Mercedes may be designing their wheels based on diatoms as well, and I actually believe that it possibly will be happening quite soon. So on to the architecture bit that came into the introduction of the talk. So diatoms here, various architectural gems from around the world here as well. The similarity in this whole idea of the structure that is aesthetically pleasing being important for giving strength to buildings as well, and some examples of that. So I've never been to the Alhambra. I keep on telling my wife that I want to go, and she doesn't take the hint, but I've now said it in public. But if you look at buildings like from the Alhambra, and you can just see, I'm not saying these people were inspired by diatoms. They haven't got a clue what diatoms were, probably. Well, they certainly didn't, because they didn't have the microscopes available. But you can see how in these architecture, these wonderful buildings, the similarity is really there. I'm not quite sure it's true of the Leaning Tower of Pisa. So here you have the Leaning Tower of Pisa, and this is a diatom. Well, you kind of, there's an obvious similarity that's there, and there you go. So one of the most iconic, I guess, for me, it was Fry Otto in the 1980s. He was the one who designed the Munich Olympic Center, and he was very big on organic shapes and took a lot of inspiration from shells and from nature. And lo and behold, he spent a lot of his time looking at diatoms and the holes and the structures of diatoms, and some of his work on diatoms and just observations of diatoms is really inspirational. But he also just looked at soap bubbles as well in a dish for the inspiration for a lot of, and he's still going into the office apparently. He and his team in Munich are still very much engaged in doing this kind of work. So it's been kind of inspirational on that kind of scale. Okay, so this comes on to we're getting there, we're nearly finished. Ernst Haeckel, he was an artist and a scientist, so he is really somebody who really did cross over. He was He could draw, he could reflect stuff that he saw down the microscope. He was the person that termed the term ecology for the first time, so he was the first one to use the term ecology. Big mate of Charles Darwin, so he was the one that in Germany at the time was really carrying on and publicizing Darwin's ideas. There was a lot of correspondence between them. And clearly a very capable scientist. What made his, he was somebody that, he studied a lot of science, so he did a lot of world trips, his big trips were to Italy and also to Ceylon as it was then, but Sri Lanka now. But he traveled the world. He also collected material that people brought back from oceanographic expeditions, so he did a lot of work on stuff that wasn't collected by himself, but he was such a renowned scientist that people would send him stuff to Jena in Germany. His most popular work, or what made him really famous, I guess, was this work, Kunstformen der Natur. It was right in the middle of the Art Nouveau movement. It's got, you can buy a cheaper version on Amazon, which is actually well produced by Presto. That is still a bestseller now. It's used by all sorts of people. Actually, the only problem is not many of the marine biology students that I come across will actually look at it, but from outside of the marine world will actually look at it. It is worthwhile having a look at. It was inspirational. He looked at dinoflagellates, so you can see these beasts that I've shown you photographs of previously. So these are a special kind of algae. These are tropical ones. So this is the one that you saw photographs of, or something similar to it. These things with these big crowns and helmets are something that was from the Red Sea, actually. And he also looked at some other glass-like organisms. These are radularians, another kind of beast from the oceans, and he was a radularian expert. So he was the top radularian expert in the world at the time, and he did a lot of drawings of radularians and other organisms, and that's great. These radularians are kind of interesting in that at the Paris exhibition in 1900, there was an architect called René Binet. He obviously worked together with Haeckel, they corresponded, but this picture here is supposedly meant to be the inspiration for the entrance area to the Paris exhibition in 1900. There's no reason not to believe it. It was inspiring for René Binet as well. And René Binet, I'm not going to go through this much, but throughout his books and throughout his literature, there's lots of the kind of stuff that I've been talking to you about that appears within his text. So that was Germany and France coming together and doing some really bright, exciting, decorative stuff. At about the same time, this is what we were doing in the UK. We were sticking pineapples on the top of buildings. It's got an aesthetic, and it's not quite as subtle, I guess. Apparently, I hadn't realized it. Well, I went onto the website to check on this. You can actually stay in this building now, so you can actually stay under the pineapple if you would like to do so. So where does this come in? So I liked Haeckel's stuff. Actually, I just would say, because I think it is important, actually, because I would say when we go back to this picture here, there was something about Haeckel. I really liked Haeckel in about 2000. Actually, I don't think much of him as a character anymore. He is a rogue. He is an absolute rogue. So in his drawings, there is an immense amount of artistic license, of which you think, it's absolutely fine, because he really brought a wonderful world to, he was the David Attenborough of his time in Germany. I mean, he was probably even bigger than David Attenborough, if you could have ever imagined something like that. He exposed Europe to a completely different world that Europe wouldn't see if he hadn't have been around. And I think that's great. But he liked everything to fit his way of thinking as well. So there's one of the things I learned in Liverpool from, ontogeny recapitulates phylogeny. It's about one of the first things I learned, and the only thing I learned in my first year there. And what basically that means is once an egg is fertilized, it goes through all of the stages of various stages of evolution before the final human is. So, there would be a a frog stage, there would be an amoeba stage, there would be what comes of well, not a bird, but anyway, kind of, and there was a set of pictures that this guy produced that actually said, well, I've taken embryos of various different stages, X number of days, and look how they are. They look. It was all fallacy. It was all a lot of rubbish. I mean, he just took pictures of embryos and said, well, this was taken from humans. And yeah, you just think, you know, you and I could do that. If I tried to do that kind of thing, I would be sacked. And so there's part of Haeckel that he had an ambition that I think was worrying. And I have to say, starting off being a real Haeckel fan, I'm not anymore. But I can't. So as a Haeckel fan, I started working together with Sarah Parker Eaton, who works in jewellery. She was also a Haeckel fan, and she created work in silver and rarely gold as well. And that was good. We started working together. And then locally, Louise Hibbard, who is a designer maker who works in wood, was also a Haeckel fan. And the first pieces of work that she did were based on this picture that I've shown you and this thing, these wooden boxes that were are really quite wonderful, based on Haeckel kind of images as well. And we came together. We didn't have any funding, but we came together. So actually, what was the connection? I bought a bottle stopper from Louise. She came to visit, and we started talking. And we said, let's work together. And so she started looking at diatoms in a new way and started producing work. And then Sarah came in and was able to put some of the embellishments on the diatoms as well. So it developed. And then we started looking at them in some other way. And then, you know, we started, you know, this is a picture I've shown you before. And then the interpretation started developing. And that was all well and good, and it was an exciting time, and we were really getting on well, and it was all thrilling. But then we got some money, I can't remember where from, but basically they came into the lab and actually started looking at these things down the microscope. And it was quite really intriguing to see, because of course they had never realized that they move, because they'd always looked at these pictures. And then under the microscope, these things actually have movement, and they kind of move quite remarkably, because, I mean, moving through water, if you're an organism that's so slow, it's cited in the literature, it's a bit like you or I trying to swim through treacle. You know, our movements would be really quite different than they are in normal water. So it was a revolution when they started trying to depict in their design and that they were making this kind of movement as well. And it wasn't very long before they got into jellyfish. Just like Haeckel did. I mean, Haeckel was a big jellyfish person. One of his key illustrations that everybody knows is this one. And so they also got into this kind of idea of, don't worry, we're just about finished, into this idea of jellyfish and movement as well. And that was great to see how that worked. But what was really exciting was that, of course, they started using the microscope for other things, and they realized that actually pollen grains, and if you look under the microscope at a pollen grain, wow, that's a whole different world that kind of opens up, but it's also based on holes, fractals, mathematics, geometry, and the geometry that is there to make sure that something perfectly does what it's designed to do. So they look at pollen grains, and they also looked at seed pods as well, and their work has evolved, and they're now looking at lots of other things. But when we worked together, we got as far as seed pods. That was good. So basically, it all kind of falls down to this, and this is a quote that doesn't come from anybody. This is where I come from. You know, I really think a scientist who fails to see the aesthetics of their science, or the artist who fails to see, or at least have a perception of the mathematical basis of the form of the subject, will always fall short in their respective interpretations of nature's design. And I guess, you know, that kind of sums up where I'm coming from and where I've got to in this subject matter. I will say, the last slide is blatant, unashamed advertising. But why not? You're sitting here and you haven't walked out yet. So with Peter here, we did produce a book on Ernst Haeckel's work that hadn't been published previously. So there was a whole series of images that hadn't been published before that we published a few years back. And you can easily get it it's published by Presto, and the same people who publish art forms in Nature. I think it's worth having a look at, of course I do. But again, some of the concepts that I gave just now and some of the background that I just gave just now into Haeckel and some of his work you'll find in the frontispiece. I mean, the only reason to buy that book is because of the images of Haeckel, of course it is. But there's some blurb at the front that we wrote where it tries to explain some of the science behind why he did what he did and put it into some kind of context of society in the time that he produced his work as well. Phew, thanks ever so much, and I will leave it. And I'm rest assured, Jane says they're going to start selling it here, so I mean, you're going to be able to buy your copies here. So, okay, thanks very much.